So let's say you want to study the effects of a gene. You can do that by suppressing or deleting that gene, and you can do that with two mechanisms, RNA interface or CRISPR. Before we decide on which one you want to use though, let's look at the mechanisms behind each. So in RNAi or RNA interface, double-stranded RNA is entered into the cell where it's cleaved into smaller fragments by an endonuclease, which is called a dicer. That can be cleaved into mRNA or siRNA, which then associates with the RNA-induced silencing complex, which then associates with the target mRNA. So if it matches perfectly, it'll cleave that mRNA, which basically suppresses the expression. It degrades the mRNA, or it can degrade the poly A tail and block translation if it doesn't match perfectly. So in the next slide, we're going to see this is the risk, and what happens here is it's just cleaving that mRNA right at that break there. Okay, well, what about CRISPR? Well, during this process, a guide RNA directs a Cas9 nuclease to the target DNA. This Cas9 then creates a double-strand break in the DNA, and the cell tries to repair that DNA in two ways. Now, for our purposes, we're just going to look at the NHEJ repair method, where basically it takes both strands and puts them right back together where the break was. That inactivates that strand, inactivates that genome, and won't be expressed. The other way, molecule-directed repair, is a way you can actually put sequences into the DNA. So CRISPR is very versatile, and you can use it in many ways, but we're not going to look at that process, because that's not a way you can degrade or suppress gene expression. So this is a animation just showing Cas9, that's the large part around there, breaking that DNA into on both strands. So which one would you want to use? Personally, I would go with CRISPR, and my main reason for that is because RNAi tends to have a lot of off-target effects, so it affects other RNA strands that you might not be looking at, so it can kind of mess with your results. You don't know exactly which, what genes are being degraded. CRISPR is very specific and has few to no off-target effects. Also, CRISPR has been improved over time, so you can use it to suppress as well as knock out genes, whereas RNAi is limited only to knockdown experience, so you can only suppress genes. RNAi is reversible, so that makes it a little safer, but the off-target effects, in my opinion, negate some of that, so I would personally go with CRISPR.